tonight's run of show, we want this to be the first part of it, be a very informal conversation amongst two friends where we're the privileged fly on the wall listening in. And then the second part of it is we want to ask all the questions, right? We want to pick Kevin's brain. Sorry, Kevin, I'm going to put you on the spot here, but we want to pick his brain. We want to ask away, and don't be shy. We want this to be engaging, and we'll call on you for the second part of it. We also have Kevin's book here. He has pre-signed them, but if you would like him to customize it, he's more than happy to do that at the end, where we'll have continued cocktails over more conversation. All right, so let's, a little bit of a background on both gentlemen. A little bit about Sean McManus. Sean McManus is a Faena Rose member. He is also the co-founder of N+D, a strategic communications firm that guides some of the world's most influential media and technology companies, foundations, and global brands on interactive content development, partnerships, and international policy. Sean was also the programs director at The Economist magazine, where he oversaw conferences and custom content globally. He has also worked at The Atlantic, launching their conference business for the digital platform called Quartz. Sean regularly advises CEOs in the area of communication strategy, thought leadership, corporate branding, and so much more. He started his career as a journalist, having written for the New York Times, New York Magazine, and numerous other publications. A little bit about Kevin Davies. Kevin Davies is the executive editor of the CRISPR Journal, and he's also the founding editor of Nature Genetics. He was also the author of Editing Humanity, the CRISPR Revolution, and the New Era of Genome Editing, for which he won a Guggenheim Fellowship in 2017. Again, we have that book here. Davies earned a master's degree in biochemistry from the University of Oxford and a PhD in molecular genetics from the University of London, and held postdoc fellowship at MIT and Harvard Medical School before moving into science publishing as an editor with Nature magazine. His previous books include Breakthrough, about the race for the breast cancer gene, Cracking the Genome, and the $1,000 genome. He's also the co-author with Nobel laureate Jim Watson of DNA, the story of the genetic revolution. Ladies and gentlemen, it gives me great pleasure to welcome Sean McManus and Kevin Davies.
All right. Good evening, everyone. It's great to see you. Thank you so much for joining us in person tonight. And as Pablo said, let's make this very interactive. So please get your questions ready, and we'll have a lot of time for audience Q&A. A lot of people ask me, how in the hell are you going to moderate a conversation about genetics tonight without a PhD? And I said, as usual, I'll wing it. So we'll see how this goes. But I'm really excited for the conversation tonight about what is really the most significant advancement in science today, and that's gene editing, and learn more about how this new technology has the promise to cure some of the world's most deadly genetic diseases. And, you know, that might not be that controversial, but there certainly are a lot of elements to CRISPR that are controversial, including the notion that we can create designer babies, which may have already happened, as far as we know. And we'll hear a lot more about that tonight. I think it should be noted that Kevin's book, Editing Humanity, is one in a series of major books coming out around this time about the CRISPR revolution, which I think is telling in terms of the impact that this technology will have on the world. It's also, and Kevin, we can talk about that, quite cosmic, given that this is happening during a pandemic, when the very methodology around CRISPR is really based on the notion of how can bacteria fight against viruses. So it feels rather cosmic in that regard. So we're thrilled to have Kevin here tonight, and thank you so much for joining us. Before we dive into the book about CRISPR, and we do have some slides that we can show periodically throughout this conversation, I was hoping, Kevin, that you could just kind of give our audience and me a bit of a sort of genomics 101 kind of explanation. You co-authored a book with someone who is the co-discoverer of the double helix of DNA. And I was hoping you can kind of tell us a little bit about kind of how we got to this point in terms of genomics.
Yeah. How long have I got? 90 seconds?
90 seconds. Go.
Anyway, thank you everybody for being here, coming out, and thanks to Faena Rose for the invitation. First flight in 15 months. Really thrilled to have the chance to come down here. So over the course of 30 years, I've written 5 books, authored or co-authored, and the 4 that led up to the one that you'll take on your way out this evening have all been about advances in genome sequencing. So we all carry DNA. We are all virtually identical at the DNA level. When you look around this room, you see this wonderful array of variation in humanity, beauty and bronze and everything in between. And we're 99.9% genetically identical to the person sitting next to you and anybody else in this room. So the amount of DNA variation, when you think about the entire 3 billion letters of our genetic code, in one sense is really vanishingly small. But those variations, of course, make us unique. They make us who we are. They control so much about us. So over the last 25, 30 years, we've seen a revolution. That word is almost obligatory in every subtitle of any book that I write. In DNA sequencing, we've gone from analyzing individual genes like the breast cancer gene or the muscular dystrophy gene or the sickle cell gene to revealing the entire genetic code of a human being. That was, of course, the Human Genome Project, a 10-year, actually a 15-year, gargantuan international project that Francis Collins, the current director of the NIH, played a very big role in, and Eric Lander, who's just been appointed as the science advisor to President Biden, was one of the leading genomic leaders in that undertaking. And there was a book in that story, not because of the quest itself, but because in 1998, a maverick scientist named Craig Venter, who many of you have heard of, I suspect, decided to launch a hostile takeover of the Genome Project. He thought he could do it faster, cheaper, and better than anyone else. And he formed a company called Celera. And so that was a very exciting and an important time. And then, as the dust settled on the Human Genome Project, Venter and others said, that was great. We've sequenced one human genome. That's not enough. That's nothing. We've got to sequence your genome and your genome and your genome, everyone's genome. We need to have that. So in order for that to happen, to really fuel and power genomic medicine, personalized medicine, we've got to get the cost of DNA sequencing way, way down. So that was the genesis for my follow-on book, The $1,000 Genome. The cost of sequencing a human genome went from literally billions of dollars at the turn of the century to today about $1,000. It truly is one of the most remarkable technology advancement stories that I think anyone can think of. So all of that's wonderful, and we can talk another time, and Robert Greene will certainly talk about what we can learn from sequencing a whole individual personal genome, in Robert's case, at birth or shortly after birth. That's one of the major initiatives that he's launched at Harvard. But what CRISPR, this new technology that just burst on the scene less than 10 years ago, what this allows us to do is now take that DNA sequence and go in and look at any gene in principle, not just in human beings, but in any organism, and change it safely and precisely in such a way that we're now on the brink of curing terrible, dreadful genetic diseases like sickle cell anemia and blindness, and a long, long list that we'll give some other examples in the course of this evening. CRISPR's made all of that possible, and of course, at the end of last year, the 2 inventors, there's still a lot of debate and discussion about who truly invented CRISPR gene editing, but the 2 inventors, Jennifer Doudna and Emmanuel Charpentier, won the Nobel Prize for chemistry for their work. So all of this seemed to suggest that there was enough of excitement in this field and plenty of ethical discussion as well to make me want to write a book, and many others, including Walter Isaacson, who has a bestseller on the list at the moment, The Code Breaker. Our books are very similar. So some of you may have read The Code Breaker or picked it up and plan to read it. It's sitting on your bedside table. If you've looked into that book, you'll be very interested in this book as well. It's two sides of the same coin.
Mm-hmm. Kevin, so for a non-technical audience, tell us a little bit about, just, in simplest terms, this notion of genetic scissors. What's actually happening scientifically to a genome when it's being cut by CRISPR?
Yeah, sure. So I've got just brought a couple of slides with me. I don't want to turn this into a lecture, and we're going to keep it very light, but I wanted to, in leading up to that, to Sean's question, just show you how immense CRISPR has become. It's almost a household term. It's an acronym. You don't even need to know what it actually stands for, although I may mention it. It's been referenced on Jeopardy! It's been one of the lead stories on 60 Minutes. This was an interesting episode. It was recorded a few years ago, and the opening scene in Editing Humanity is this. This is Feng Zhang at the Broad Institute holding up a little vial of CRISPR with Bill Whitaker. They had to reshoot this scene because originally this episode was filmed with Charlie Rose in the interviewer chair, and then he lost his job, and you all know that story. So they had to refilm the entire episode with Bill Whitaker sitting in for Charlie. And of course, it's been at the megaplex. CRISPR took on Dwayne Johnson and lost, because of course everyone loses to The Rock. And here's the Nobel Prize for Chemistry, shared by Emmanuel Charpentier on the right, and Jennifer Doudna on the left. Usually they receive their Nobel Prizes at this glamorous banquet in Sweden, of course. Because of COVID, they had to do this in slightly different circumstances. So Emmanuel, who works in Berlin, picked hers up at the Swedish Embassy in Berlin. And Jennifer is literally in her backyard in Berkeley getting her prize from the Swedish attaché or something like that. Mm-hmm. You asked about CRISPR, and we don't need to obsess over this diagram, but CRISPR is it's a small little funny, strange, weird sequence, DNA sequence or set of sequences in bacteria. For about 20 years, a very small, tiny band of scientists who are not famous and you have never heard of decided they were fascinated in CRISPR. There was something peculiar about the pattern of these DNA sequences that made them think DNA doesn't just sort of form these repeat structures by accident. Evolution must be telling us something. There must be some purpose to this. But for 20 years, these scientists just couldn't figure it out. And to cut several chapters short, what they found out is that bacteria have a way of defending themselves against viruses. They have to, otherwise they would be dead and life on this planet would not exist. And the way they do this is to develop a sort of a storage system. It's like a little bacterial genetic filing cabinet in which they store DNA fragments from the viruses that they've been able to vanquish. And they store it so that they can tell apart a viral DNA trying to invade a bacterial cell from its own DNA. You don't want genetic scissors running amok, killing your own DNA. That wouldn't be too good. So you need to tell friend from foe, and this is what CRISPR is. And so in the early 2000s, several teams of scientists figured out, sort of pieced together what this CRISPR array looked like. Okay, that was all very good. This is a viral defense system. This is a bacterial immune system. But it was Doudna and Charpentier in 2012 who, building on many other important pieces of work, we stand on the shoulders of giants and all that stuff, they turned the basics of that system into a genetic scissors that, in the way that bacteria use CRISPR to program an attack against viral DNA, incoming, invading viral DNA, Doudna and Charpentier and their teams turned that around, made a couple of small tweaks and said, we've now developed a scissors that can target any DNA. We specify what that DNA looks like. So CRISPR, when people say we're going to CRISPR this gene or CRISPR this organism, we're just talking about two components. We have the CRISPR, a small bit of genetic material that we spell out the A's, C's, T's, and G's, the genetic alphabet, to target and line up with a specific gene that we want to target. It might be the hemoglobin gene for sickle cell disease, or a gene that, when mutated, causes deafness or cancer. And then there's an enzyme that does the actual cutting. Think of it like a Pac-Man or genetic scissors. That's the term the Nobel Committee used. Mm-hmm. And put these things together, you have a homing system and you have an enzyme. And many scientists have built on what Doudna and Charpentier did. They've got this to work in every organism, including humans, and that's the ground foundation now to a biotech industry, a number of public companies, and they're all off and running, launching clinical trials to tackle a growing list of genetic diseases.
Yeah, so we definitely want to hear exactly how those applications are going to work. One thing I just want to mention very quickly is the origin story that you cite around the University of Alicante in Spain and Francisco Mojica.
Yes.
What happened there in Spain in the 80s?
Well, we visited Alicante, the University of Alicante, as part of the research for the book. And these are the salt lakes just south of Alicante in the southeast corner of Spain, where the climate it's a little drier than here, but it's beautifully warm. And these are lagoons where they concentrate salt out of the Mediterranean, and just off camera are huge mountains of salt that are used for both culinary and industrial purposes. There is a microbe that lives, that thrives in these increasingly salty conditions. And Mojica was one of, if not the first scientist who uncovered these CRISPR DNA sequences, and he thought that these microbes must have evolved or must be using CRISPR to survive in this incredibly salty environment. It was a lovely theory. It was completely wrong, but that it fueled a fascination with him. And right up to the Nobel Prize, when it was awarded last year, he was very much in the running. Perhaps not quite the favorite of Doudna and Charpentier, but the Spanish media, they made him a rock star.
Yeah.
And when we visited him, we after this tour, we took him for just a beer before we went back to our hotel, just to ask a few final questions after the tour of the Salt Lakes. And said, so what's it like? You're such a star now. You're almost a household name in Spain. You've gone from this obscure microbiologist in not a major league university into this household name. You're about to go on a month-long lecture tour of Australia. You must be loving this. And he sort of put his beer down and he sort of leant forward and whispered, I hate it. I hate it. I just want to go back to my laboratory. I don't want to be troubled by all this stuff. I just want to go home to my wife and have dinner. It was very sweet. It was not the answer I expected, but it shows that basic research, it takes all sorts, and they do it not because they dream of fame and fortune, just because they found a little weird sequence in a bacteria and they had to know what it did.
Yeah. We'll see if he gets addicted to the fame after a month of touring.
Yeah.
Kevin, can we fast forward to 2018 when it was revealed that a scientist in China had potentially implanted 2 genetically modified genes into an embryo?
Yeah. So all the CRISPR applications that we've just very briefly touched on so far, treating genetic diseases, trying to do this sort of form of gene therapy where we would go in and edit the gene of a child or an adult with blindness or hearing loss, deafness, sickle cell disease, other blood disorders, liver diseases. The list is growing on and on. There's nothing terribly controversial about that. Perhaps the most controversial element of that is that gene therapies, the ones that have been approved so far, are outrageously expensive. They're upwards of $2 million. And that says something, and you all know that we all have a sort of a love-hate relationship with our healthcare system in this country. We all think that American healthcare is the best in the world, and God damn it, I'd swear to it. But then why are our drugs so ridiculously high? Why is it cost? Who the hell is going to afford $2 million for a genetic therapy, for a one-time injection? Now, and of course, the companies, the biotech companies, will say, well, we developed this drug. We use a little bit of taxpayer-funded basic research, but if we don't get a return on our investment, we're not making any more gene therapy drugs. So there's this sort of tension between do we appease our shareholders or do we serve our patients? And sometimes I think that gets a little bit out of whack. But put the pricing to one side. Maybe we'll come back to that later on in your questions. But there's nothing too controversial about saying we've got a new gene editing form of gene therapy and we want to cure you or your family or your child because of a genetic disease. But what He Jiankui, a Chinese scientist, let me just quickly scroll to his photograph because I have it coming up. So here's He Jiankui, a young 30-something Chinese scientist who trained in America. He did his PhD at Rice University in Texas. He did 1 year of postdoctoral training at Stanford with a gentleman named Stephen Quake, who is a very well-known, or at least in California, very well-known scientist entrepreneur who's co-founded many publicly traded biotech companies. So this guy was getting a crash course in genetics, even though he was a physicist by training. He was lured back to China with big grants, and he landed at a university in Shenzhen, just across the border from Hong Kong. And everything's going well. He launched a DNA sequencing company. He was flying high. He didn't need to be dabbling in CRISPR or gene editing. But somewhere along the way, he sort of decided to fly a little too close to the sun. He felt that he wanted to do genetic engineering, not of grown patients or children, but he wanted to try editing the DNA of a human embryo. And why is that so controversial? Because when you make an edit in a gene in that one cell embryo, then of course as that cell divides 2, 4, 8, 16, 32, you can do the math, that edited bit of DNA is going to replicate and spread across through every cell. So when the baby's born 9 months later, that baby is fully stocked with that edited gene and is then in principle able to pass that gene on to their children and future generations. So this is literally changing the human gene pool, okay, albeit one child or 2 children at a time, but this is man kind of playing God. And some may kind of balk at that sort of phrasing, but I really think that's what it is. And he decided he wanted to do this for a specific disease, a well-known disease, of course, HIV. Now, there's a gene that encodes the receptor that the HIV virus locks onto to enter our cells. And about a small percentage of you in this room, you may not know it, but you are carrying a naturally occurring mutation in this receptor gene that makes entry of HIV impossible. So you are naturally resistant to HIV. This is a well-documented mutation. He-Jian Kui, I'll call him JK from now on, JK wanted to mimic that deletion. There's 32 missing letters of genetic code in these human embryos because HIV is still a major public menace in China. There's still huge stigma about it. And he felt he wasn't just doing this for the families concerned that he was enrolling in this trial, but this was about raising Chinese science. This was about elevating not just his own group and his own reputation, but about really putting Chinese science on a portal. He was going to be the first scientist in the world to deliver gene-edited babies. And this all came to a head in Hong Kong at a conference in late 2018. There's many chapters in the book on this historic episode. Here is his email that the New York Times published to his former boss, Steve Quake at Stanford, just announcing the pregnancy. He confided in many American scientists, but then he swore them to secrecy. Please don't breathe a word of this. He wanted to be the first to announce this. And just on the eve of this Hong Kong conference, he did in a bizarre, he announced it because he'd been scooped. He'd been scooped by this brilliant science journalist, Antonio Regalado, who's with the magazine MIT Technology Review. He'd met JK a month before. He got wind of what he was doing. He couldn't put all the pieces together when he met him, but he went back home to Boston, did lots of Google searches in the wee hours of the night, and on one, I think it was a Sunday morning, he finally found the retroactive posting of a clinical trial under JK's name, mentioned the gene, it showed DNA counts from pregnant women, and so he burst this out in a story that was posted later that day saying CRISPR babies are imminent. He didn't know that they'd been born, although they had just been born, but he had all the other pieces of the story. It was a brilliant piece of journalism. That was his exclusive story just on the eve of the conference. Mm-hmm. And so suddenly 500 journalists from all across Asia and the world, this was like attending a rock concert rather than a scientific conference. There's usually maybe one photographer in the front row, you know. This was absolutely insane. You can see the press scrums. It was like David Beckham, having to explain why Inter Miami have lost another game or something like this. And the ethicists, the scientists community, as the word got out of what he'd done, they just came down on JK like a ton of bricks. He had no right to be doing this. He may have forged documents. He didn't have enough control of the editing. He should have discussed this with the science community. You just could make, as Ed Yong in The Atlantic did, a list of a top 50, not even a top 10 list, a top 15 list of reasons he shouldn't have done this. And the most important reason, I think, that was stated at the conference was from a senior Chinese the elder statesman of Chinese bioethics. So in a way, a friend of Dr. He, of JK. How could JK and his team change the gene pool of the human species without considering the need to consult other parts of the human species? I mean, yeah, absolutely. So that, for me, I think sums it all up. So JK, I think this is my last slide on this little section, left Hong Kong. He was escorted back to Shenzhen. He was immediately put under house arrest while the Chinese authorities investigated him. I suspect there were some senior members of the Chinese regime that knew exactly what he was doing. I don't think he was the rogue that he was widely portrayed to be. And a brilliant young New York Times staffer based in China. Everyone went looking for this guy, and she played a hunch. She went to one of the guest houses on campus where people are put up for conferences and things, and she saw this lonely figure hanging out on a balcony and zoomed in, and sure enough, it was him. But she couldn't get any closer because there were about 12 armed guards surrounding him. 6 or 9 months later, he was jailed. There was a trial. He and 2 of his collaborators were sentenced to, he was sentenced to 3 years in prison, so he's halfway through that term. And there have been big reports from leading scientific groups led by the National Academies of Sciences that have tried to say, what can we learn from this episode and what are we going to do going forward? And so hopefully this won't happen again, or if it does, it will happen under very controlled circumstances.
Well, so let's be honest, though. I mean, you pointed out that this technology is actually affordable. It's actually comparatively not that difficult to use. You've used a term in the past called CRISPR cowboys. The notion of rogue scientists, going off to an island off the coast of Malta and doing this is actually not.
It's not completely out of the question.
Yeah.
So even though a big report was published late last year from a blue ribbon panel of scientists digesting everything that had happened in the past 2 years, precipitated by this fiasco, and said the only circumstances we think hereditary genome editing should be potentially allowed in the future is if, let's say there's a couple and both have a very serious genetic disease, such as cystic fibrosis or sickle cell disease. Both of these are recessive diseases, so if you have this disease, by definition, you are carrying both copies of the gene in question are broken, both are faulty. Normally, when you offer a family the chance to have a biologically healthy child, even though there's a family history in the family, of a disease in the family, you can offer a form of IVF called pre-implantation genetic diagnosis. So you can do in vitro fertilization, you can gather the newly fertilized, the newly grown human embryos in a petri dish, and you can do a little DNA biopsy. You can just take 1 or 2 cells off each of the 5-day-old embryos, look at the DNA, look at the gene in question, and you can label each embryo from 1 to 8, has the gene, doesn't have the gene, has 1 of 2 copies, and on that basis decide which of those embryos you're going to reimplant to give that couple the best chance of having a healthy child. But if both members of the couple, both partners, have one of these serious recessive diseases, then that option isn't available. There are no healthy genes at the specific gene in question that you can pick from. So in that scenario, maybe gene editing might be permissible. But you do the math on the back of an envelope. What are the odds that 2 CF patients or 2 sickle cell patients, they're not super rare diseases, but they're still relatively rare genetic disorders. What are the chances that they're going to marry and want to eschew adoption or some other method to have this biologically healthy child? So we're probably talking a handful of cases in any given year at best, and probably there are more experts writing these reports than it would actually apply to. But I agree with you, Sean. That doesn't stop, and even though we have these blue ribbon committees writing and issuing these reports on impressive stationery, it doesn't mean that some entrepreneur isn't going to say, right, I'm going to go off to Macau or somewhere in the Middle East or wherever. I don't know what you've got against Malta, but maybe Malta, and say, I'm going to launch a CRISPR clinic. And we know there's interest because in the days after JK, even though he was under house arrest, he was getting I don't have it in this deck, but he received an email from a fertility clinic somewhere in the Middle East, I think it was in Dubai, saying, we were fascinated by your work, Dr. JK. Please tell us, provide training materials so that we can offer this to our clients.
Yeah.
So we know that there was some pent-up interest in replicating this. Maybe they're laying low now because they've seen how severe the backlash has been.
Okay. Well, on the more legitimate side of things, so get your questions ready. We're going to talk a lot about all the different applications for CRISPR technology, which are far-ranging, from agriculture to controlling mosquito populations. I want to ask you about that because we live in Florida and summer's coming. But the basic commercial applications as they exist today. I had the occasion to meet Sam Kulkarni, I told you, who's the CEO of CRISPR Therapeutics. There's a number of others. Can you give, for some of the people in the room who are private equity investors, venture capitalists, and who might be interested in actually putting investment capital towards some of these commercial applications for CRISPR, can you give us a bit of the landscape that we have now?
Yeah, so in the last 5 years, there have been, I would say, I think 4 CRISPR-related companies are now publicly traded, and there's a whole slew of new, very handsomely financed startups getting Series A and B rounds that I think are on the verge of going public. CRISPR Therapeutics is, partly because it's got CRISPR in the name, is perhaps the best known, because they led this clinical trial with this patient. This is Victoria Gray on the slide. She was the first American sickle cell patient to receive a CRISPR gene editing therapy, and it worked. And boy, it worked. People don't want to use the cure word, but they're having a hard time not doing it because she's now 18 months, almost 2 years now, past her therapy, which was, the trial was sponsored by CRISPR Therapeutics. Vertex Pharmaceuticals is now, I think, taking over, if not kind of the lion's share of that work in a partnership. But what they were trying to do in Victoria's case is not fix the actual sickle cell mutation. They said, let's leave that alone. We have another idea that actually works better with the CRISPR, the pros and cons of actually using CRISPR. There's another gene that we all carry that makes a form of hemoglobin called fetal hemoglobin. So when you were in your mother's womb, you were happily expressing this gene, and it worked great. And then shortly after you were born, you naturally, as we all do, we shut that gene down, and another gene is expressed to take its place. But the fetal gene, it's called gamma globin, it's sitting there, it's perfectly intact, it's perfectly functional. It's just that we don't need, it's almost like a globin on steroids. We don't need it because we're adults now, we're growing up. But it's sitting there, so if we could reactivate it, just kind of wake it from its slumber, it will, in principle, replace the broken globin gene that's messing up our hemoglobin and causing these sickle cells. So that's the HbF that you see in this slide. And when nearly 50% of the total hemoglobin in this woman's red blood cells are this new reactivated fetal hemoglobin, that's an amazing result. You probably only need 15 to 20% to actually achieve a meaningful cure. So they have to now extend this, of course, to other patients, other disorders. So this is not yet FDA approved, but the early results are great. Other companies are Editas Medicine. That was the first company that went public. These are pretty much all in Boston. There's another company called Intellia. They're all looking at sometimes the same diseases, sometimes slightly different diseases, all using CRISPR. And then, maybe I'll go to it now. There's a fourth company that I want to mention, which I mentioned, just bear with me while I just quickly fast through these slides, but it gives me a chance to talk about it. Here we go. So there's a scientist, a chemist at Harvard University named David Liu, and even though CRISPR has already been awarded the Nobel Prize for chemistry, I'm going to go out on a limb, and I think that David Liu, maybe 10 years from now, certainly would be one of my odds-on favorites to win a Nobel Prize for medicine. He's developed over the last 5 or 6 years some people call it CRISPR 2.0. It's a riff on CRISPR. It uses some of the CRISPR machinery, but a couple of different elements as well. But it allows him to make gene edits in a safer fashion than CRISPR. When you do a CRISPR experiment, you actually cleave the double helix completely, and then you let the cell naturally repair those broken ends and stitch in the correct gene or the healthy gene that you've administered. That all happens pretty naturally and pretty seamlessly. But it's still a very abrupt thing to do, to completely sever the double helix. In David's system called base editing, you just nick one of the strands of the double helix. So it appears to be a safer process. And while he can't fix every genetic mistake, in the case of sickle cell disease, he can change the sickle cell mutation into a very harmless, a different letter that is completely harmless. And so these are just 2 newspaper headlines from the last couple of months, because this guy's on an absolute tear. And indeed, the photo on the left, taken from January, is advances in treating a rare and awful genetic disease called progeria, Hutchinson-Gifford progeria, which is a form of premature aging. That child in that photo is probably, 4 years old, but teenagers with this disease look about 80, and they die young, of course. Working in mice, you have to do experiments in mice and animal models before you start doing CRISPR experiments in humans. That's common sense. That's how clinical trials work. He's pretty much cured progeria in a mouse model, and that sets the stage for treating these patients in a clinical trial. And just 2 days ago, beautifully timed, I think they must have known that we were having this discussion, there's David sitting in his office at the Broad Institute, where they've just published in Nature, my old magazine, a cure for sickle cell disease in mice. Not doing the fetal thing, the strategy that we were just talking about. This is going in and actually repairing the single base, single letter of the genetic code that is broken or faulty in sickle cell disease. David would be if he was sitting here, and I hope you bring him down at some point, David would be the first person to say, we are light years away from curing this disease. He's a meticulous scientist. This has got to be confirmed and replicated and done and extended in other animal models, and then you very cautiously commence a clinical trial. He has, but that's obviously in the planning. His company is called Beam Therapeutics. They went public about 18 months ago. Beam stands for base editing and more. They're also based in Cambridge, Massachusetts, which is the mecca of biotech companies. And I've got very high hopes for what Beam is doing for sickle cell and other diseases as well.
Is that ironic that the previous slide had David with an Iron Man
He's
sculpture?
a big superhero geek. He's a brilliant photographer. I mean, this guy can do it all, really.
Yeah.
But when he was the youngest chemistry professor in Harvard history. How old was he? 26 when he joined? He did his PhD, and then they said, David, you don't even need to do a postdoctoral fellowship, which is what I was trying to do. Yeah. We're just going to promote you. We're just going to bring you in as our assistant professor.
Okay.
So he's ridiculously young. So, Harvard's, like most Harvard departments, it's just stacked full of Nobel laureates. So he wasn't getting the best students. So he would, come recruitment night, he would sort of, make these big superhero posters and just show a sort of a more flamboyant side to pull in some of the cool kids. Yeah. And it worked.
Well, CRISPR might lead to superhumans at some point.
Well, we could get there.
So then we can talk about that. But what about, what about cancer? What other big diseases are kind of in the pipeline, that you've seen already work being done?
There is some work going on in cancer, but it's more in the sort of the immunotherapy realm. And I think the tricky thing with cancer is it's not just one disease. Right. It's hundreds, if not thousands, of different diseases. Yeah. So it's not as easy to say, CRISPR is going to be the cure for breast cancer or prostate cancer or anything else. Rest assured, there is work going on, but I think that's a slightly longer road ahead. And so some of the companies that I just mentioned, if you go on their websites and look at their pipelines, I'm sure you'll see certain forms of acute leukemia or something like that. So you can go in and see what they're doing.
So Kevin, what about the applications for CRISPR around agriculture? And I heard an interesting interview with you talking about, kind of reframing the debate about GMOs and what does it mean to be genetically modified, which there's a lot of misunderstanding about that.
Yeah.
But what is currently underway, and what is the potential promise of CRISPR in terms of agriculture?
There's huge promise. Indeed, people like Doudna, the heroes of this field, would say that for all of the hype and excitement that treating human genetic diseases is getting, it's really the work in agriculture and fixing our crops that is perhaps the most important, because we're not going to be around on this planet with global warming and everything else unless we make our crops and our plants more resistant. A few years ago, the New York Times had it was a piece or a series by Amy Harmon about your precious orange crop down here, which is under assault. And think of any fruit or vegetable that you care to name, oranges, bananas, whatever it is, there's some bacteria, virus, pest, bug, drought, there's something, it's under a fungus, there's something under attack. So without giving any specific examples, CRISPR is the same way that we can target and fix a gene in human cells, we can go in and fix a plant gene and make it increase the activity of an enzyme to kill off a bacteria species or something like that. So the theoretical promise is great. The first sort of field trials are going on, but you're right. There's a lot of education that still needs to be done, not just amongst the public, but amongst regulators even, who. A lot of people here probably avoid GMO. If you go into your local supermarket, you probably see a non-GMO sign, and you think, oh, thank God for that, I'm going to reach for that. I would eat a GMO food in a heartbeat. There's nothing harmful about it. It's been genetically modified, sure, but anything that's been put in is just a DNA sequence. You're going to digest it. It's fine. So there's a lot of, I think, information. We have a stigma about GMOs that we have to overcome. But so there's a bit of an uphill battle for the CRISPR community to convince a skeptical public that, well, why is CRISPR safe when I'm a little nervous about GMOs? Well, CRISPR is sort of a more fleeting, transient form of genetic perturbation. When CRISPR goes in, you're not leaving anything behind. You're going in with this scissors, you're making the tweak or the fix that you want to produce the desired property, and then it's gone. And the fix that you're making is exactly what plant geneticists at the big ag companies, the Bayers and the Syngentas and the Dow DuPonts, which now called Corteva, they're trying to engineer these variants. But until recently, they've had to do this by bombarding plants with gamma rays and X-rays to induce mutations and then breed these things for generation and generation and hope that one day, bingo, you get your whatever your property you're looking for, there's the resistant crop, and now we can start to play with that. CRISPR is the technology that allows you to do that rationally, because you design the gene that you're going after. You're not just sort of waiting, hoping, praying that you throw a lucky, I don't gamble, but you throw whatever the lucky dice are, and your radiation produces the genetic tweak that you're after. So there's nothing to worry about. And with plants, of course, they take a long time to breed, so they'll be very carefully studied. But if we don't do anything, fresh morning orange juice, it's going to be a thing of the past.
Yeah.
I mean, we're in trouble here.
Yeah.
So we've got to use the tools and the ingenuity that we have to try to fix things.
Yep. 9 billion people on the planet, soon enough, right? Do we have any questions at this point? We have one here.
Uh-oh.
Thanks. Just real quick with what you said with GMO crops, right? I agree with what you said that if you're physically editing the DNA, there's nothing wrong. But there's companies like, I guess Bayer, who bought Monsanto. Yep. Isn't wheat these days engineered to withstand more pesticides like Roundup? So it's kind of soaked in Roundup.
Yeah.
So isn't that kind of problematic?
Yeah, I think there's. I'd be out of my depth if I tried to get too deep into the GMO state of play. But so I'll go back to what I said, which is that even if there are certain scenarios where you don't like what GMO is a big field. So there may be some GMOs that are perfectly fine because they're in your grocery store and you're going to eat them, and then there's other applications that you may have more environmental concerns about. But regardless, CRISPR is it's a sort of a phantom technology to go in and make a rational tweak to the DNA, and you can do it, in a week or a month. Versus right now, there are scientists spending years trying to, engineer bug-resistant bananas or tomatoes or whatever. So I think they're turning increasingly to CRISPR as the way to really speed up and rationalize this work.
I guess when it just comes to ethics across the board, I guess that's the biggest piece of everything.
Yeah, so they're trying to, and I think some of the regulators have made the distinction between GMOs, which do typically leave some sort of gene signature behind, and CRISPR-edited plants. So you can conveniently sort of separate them. And when CRISPR modifications are lumped in with GMOs, then you have a lot of problems. So. Can we use the mic? I think because people are streaming in, so.
Sorry, I'm only like 1 week into reading all about CRISPR. What's your view on Josiah? I can't pronounce his last name.
Yeah.
Something. Essentially being like a CRISPR cowboy.
Yeah.
And this enormous amount of people in the U.S. and globally who are trying to modify their own DNA. Sounds like a lot of it is for actual good purpose, people with terminal illness, but other people just trying to maximize their body and their performance.
I did not give the biohacking community a lot of airtime in my book because I'm very skeptical about what they're doing. One of the reasons, so let me just backtrack a minute. One of the reasons CRISPR has become this household term and has won a Nobel Prize 8 years after the paper was published, which is almost a record, is it's so easy to use. You don't need state-of-the-art $1 million instruments from Illumina or one of these big companies to run a CRISPR experiment. You need almost no hardware at all. You just need some ingenuity, a couple of reagents, a little tube containing the stuff, and some DNA sequences, and there's all kinds of programs online, and you can order these reagents, and they're shipped overnight, and you're off to the races. You're doing a CRISPR experiment. So this is why there are some people in this sort of biohacking community that have decided to take it on themselves. They feel empowered, and they feel no one's going to stop us, no one should stop us, we're going to inject CRISPR and see what happens. I don't think they've fully thought out what it is they're trying to do, so there's a bit of sort of showmanship and publicity, and so I think the main reason I didn't really spend a lot of time on it is I didn't want to give additional airtime. These guys have had plenty of airtime as it is. There was a 4-part Netflix series that aired a year or two ago that was all about these guys who were trying to inject CRISPR in their garage or turn their dogs green. And my fear was that giving any exposure to this potentially tarnishes the amazing work that people like this are doing. And when JK presented his results in Hong Kong 2.5 years ago, the whispers around that packed auditorium were, holy cow, this is going to ruin the whole CRISPR industry, what he's done, because somebody in Capitol Hill or someone is going to want to regulate the heck out of this now. Because then they're not going to understand the differences between doing hereditary embryo editing and therapeutic editing in adults. Thankfully, that hasn't happened, and I sure to God hope it doesn't happen. So I hope that, you said it's a big army of people. I don't get that sense, but maybe you have other sources.
Just when you watch the Netflix.
Oh, yeah.
It sounds like everyone's doing it. I'm not.
He gets a lot of attention, but I think there were really just 2 people that series focused on.
Well, Kevin, what are there precedents for how the regulatory frameworks might play out?
I don't think anybody. I mean, I think he's coming under some scrutiny, but I don't think he's done anything terribly
illegal,
wrong that I can think of. So,
yeah.
And he's a well-educated guy, and so I think he does sort of know what he's doing. I'm not trying to disparage him.
Yeah.
I just didn't want to give it a lot of airtime in my book.
Well, it is kind of a terrifying proposition that everyone's just running around injecting themselves.
Yeah, I mean, if
with
you've got a
CRISPR.
genetic disease or there's a good reason that you want to get interested in CRISPR, then talk to these companies. I mean, there's going to be more and more of these trials getting underway for the right reasons and performed and monitored the right way.
Good.
Yeah, I have a couple of questions. One, whatever happened to the CRISPR babies? That's one. Two, knowing that nothing is 100% and things can always go wrong, especially in medicine. What are some of the things that can go wrong with CRISPR technology? And then, looking way into the future, into the sci-fi movies, if we're going to create a population of people that really don't expire, what is the infrastructure that we're creating to support these people, such as you say, all the GMO food that we need? And I always think of this movie when I was growing up, which is Soylent Green.
Yeah.
Do you remember that?
I do.
Soylent Green is made out of people.
Yeah.
Because there isn't enough food on the planet for everybody, so then the rich get immortalized, and then all the poor get mealed up into these pancakes of Soylent Green. So those are my three questions.
That was three questions, yeah, I was going to say. So what happened to the CRISPR babies? We don't know. We don't know. So I put this slide back on because these are the, when he presented in Hong Kong, on the slide in the back of this picture, these are the DNA sequence traces of the two embryos. All we know is that he went on YouTube and said their names are Lulu and Nana. There was a third CRISPR baby born 6 months later. We don't even know the name of that child. So they're about 2.5 years old now. We know their nicknames. I don't even know if that's their real name. Nobody knows. Well, somebody knows. Somebody in China knows. But nobody, to my knowledge, has even kind of anonymously said, here they are, here we've studied here's a report on their medical status, and so on. So I hope that happens. I mean, since he's done this, the genie's out of the bottle. So somebody should be presenting the results of this work while preserving their privacy, if that's important in China. I presume it is. Fine, but let's at least see their results so that the whole scientific community can better understand and advise and help their Chinese colleagues. So that's one. So it's sad, but what can we do? CRISPR is not completely 100% safe. No CRISPR aficionado would claim it to be so. One of the challenges with CRISPR is that when you give this genetic scissors, you program it to target a specific gene. And of course, a human encyclopedia of DNA in a cell is vastly more complex than a tiny viral genome, which is what CRISPR was designed for in nature. So when you program CRISPR, there's a statistical chance, the odds are low, but there's a chance that the same sequence could crop up in an unrelated part of the genome, in another gene, or maybe not in a gene, maybe in some junk DNA. But this scissors could cut where it's supposed to over here, but then maybe over on chromosome 8 or whatever, over here. You don't want it cutting on chromosome 8, that wasn't your intention, but it's going to cut anyway. That wouldn't necessarily be good. And the history of gene therapy, which I talk about in one of the chapters in the book, is not full of, but it has some very prominent examples of children who've fallen ill or developed cancer. And of course, the famous case of Jesse Gelsinger, who died in 1999 in one of the first big gene therapy trials conducted at Penn. Not because of CRISPR, CRISPR hadn't even been invented at that point, but because the dosage, the virus, triggered this massive immune reaction. So there's still a lot of work, basic research that is still being done to make CRISPR completely as safe as possibly can be, but also working on the delivery vectors, the viruses or the non-viral systems that we use to coax the CRISPR machinery into cells. So that's your second question. And the third was, I didn't know if you were meaning about CRISPR being used to increase longevity in a sci-fi concept or food. We've already talked about food.
Well, the idea that we're all terminal at some point. So if
Yeah.
one thing doesn't kill you, then something else
Yeah,
will.
yeah.
So if we're creating a population of people that with the genes and everything in the end is genetic.
Yeah.
And so at some point we'll be curing cancer with this.
Yes.
So if we're creating this, theoretically immortal species in a way, how are we going to sustain it?
There's nobody.
I'm, of course, looking way to the future.
Yeah. I don't think we're creating an immortal species. I'm sorry to disappoint you, but sadly, I don't think that's still the provenance of science fiction films. But some of the genes, as you say, if we can fix genes that cause cancer, or maybe Alzheimer's disease one day, or about heart disease, one of the biggest killers of people on the planet. There's another company I should have mentioned in that little roundup I did a few minutes ago called Verve Therapeutics. They've just announced their IPO. Verve Therapeutics is another Boston-based CRISPR gene editing company, but they're looking not at rare genetic diseases, but they're looking at heart disease. So their potential audience is much bigger. Now, if you have a family history of heart disease, you may, and some of you probably are already on a statin or some similar type of medication, and those have been wildly successful drugs, occasional side effects, but wildly successful, and you may be content to just continue on that. If someone said, hey, we could potentially go into your liver and edit a gene that might just lower your cholesterol, that's a pretty intense kind of therapy, but that may be on the cards one day. So Verve Therapeutics, that would be one worth looking at. So I think piece by piece, we'll see incremental increases in longevity because we're going to start knocking out some of the most notorious disease genes that impact our longevity. But there isn't going to be some magic thing where we're suddenly going to see a lifespan of 150 or something.
This is more of a mechanical question. In the CRISPR gene therapy, how does it replicate enough to make a difference on a gene illness level? Also, how does the cell not get lysed from the mutation?
Yes, two great questions. It depends on the disease. So in the case of Victoria Gray, the sickle cell patient, they do a sort of a traditional chemotherapy stem cell transplant. So the patient has to undergo chemotherapy. You take her, before you do that, you take her healthy stem cells, you edit those, and then you filter them, grow them, make sure they're kind of happy and ready to go, and then you infuse them back. So if it's done well, and they can find their perch back in the bone marrow, like a traditional stem cell transplant, they'll be fine, and they'll continue to grow and pump out and deliver these healthy red blood cells. So that's a slightly special case. But whether it's muscular dystrophy or some blindness, it's all about delivering enough of the CRISPR to the cells or the tissue or the organ that's affected. You're not trying to do this throughout the whole body. So in Editas's case, they're treating a rare hereditary blindness. So they're literally injecting the CRISPR stuff just behind the retina, just to attach or insert into the photoreceptor or whatever the cells are to make the fix. You don't care about any other cells in the body. So one of the reasons that disease is so appealing is it's a serious disease, of course. Anyone would love to fix and cure blindness. But the number of cells that you have to fix is relatively very small. So that, from a mechanical standpoint, that makes life quite relatively straightforward. If you're trying to cure a disease like spinal muscular atrophy or Duchenne muscular dystrophy, they've already shown in a whole bunch of Nature and other journal papers that they can make the requisite genetic fix. But it's going to be delivering that into the muscle. Think of all the millions or trillions of cells or fibers that have to be, each of them have to be fixed, because that's not going to grow and spread automatically. So each disease comes with its own sort of sets of challenges, I think. But, and the second part about lysing the cell, the CRISPR is, it's going to have a natural sort of life cycle, a natural turnover. So that the enzyme that actually does the cutting, once it's in, eventually it's going to sort of break down. It's only going to hang around for hopefully just long enough to make the cut that you've pre-programmed, and it's not going to stick around forever because there's always turnover in our cells. So that doesn't appear to have been a big problem so far. Great question,
and then we can continue over cocktails. Thank you. I'm a little bit confused with the story of the Chinese gentleman that's still on the screen.
Yes.
And what's leading to that is you mentioned that 99.9% of our genetic code is the same. And then that little last percent is what it actually expresses variation in humanity. That variation is these diseases themselves that we're trying to cure. And so in the event that he edits one mutation that I don't have, how is that him fundamentally changing the genetic code beyond the variation that's already expressed in humanity?
What he was doing, so Lulu and Nana, the edits that he's made are present pretty much throughout, in every cell in their body. There's an asterisk behind that statement, which I won't get into, but let's just, for the sake of argument, say it's present in every cell. So when they have children, that edit that JK engineered is going to be passed down to their children. And so that could spread. And he tried to mimic a naturally occurring genetic variation. The one that I said earlier on gives this HIV resistance. It's a missing piece of 32 letters of genetic code. And some of you have that variation in your DNA. And it's harmless. It doesn't have any side effects that we know of. But it has this weird thing that it prevents HIV from entering your cells. So he was trying to replicate that, and he failed because he just didn't really know what he was doing. It goes back to the biohacker question. Almost anybody can do a CRISPR experiment, and he just sort of cavalierly said, I've got no training in CRISPR, but I'm going to edit the DNA of these two human beings. So these are walking human guinea pigs. They didn't ask for this. And we don't know the effects of the mutation that he engineered because he botched it. And the variants, the deletions that he's engineered in these two twins have never been seen before in any human being. So he hasn't, even if he'd beautifully mimicked the naturally occurring mutation, people would still be up in arms about what he did. But he didn't do that. So that was one of the big, on my top 15 list of problems with this experiment, that was one of them. That we don't know the health effects that these mutations are going to have on these kids. So to the question, what do we know about the babies? That's why people want to study these twins, or at least monitor them, not study them in a kind of you're a walking experiment, we want to study you, but we want to make sure you're okay. And if this experiment has triggered some sort of cancerous event or something, we, the world, we owe it to you. We've not served you well by letting him do this, and so we want to bring everyone's collective wisdom to bear to make sure that you don't suffer any really untoward ill effects from this. So the gene pool analogy that I gave, that we're getting into science fiction a little bit, because you could say, well, who cares? I mean, how long would it take for this to spread? And it would take centuries, of course, but I'm doing a lot of reading at the moment. We're talking about sickle cell. Think about how sickle cell spread. The sickle cell mutation arose in Africa about 7,000 years ago, not because God or evolution said, oh, sickle cell disease, this is cool, because the first patient, the first person who spontaneously started walking around the savannah with a sickle cell mutation, one copy of it, that person didn't get malaria. So that person lived to reproductive age when all of his or her friends and family were, dropping like flies, no pun intended. So the sickle cell, that one mutation spread, and it spread. And over hundreds of years, it spread so much that suddenly carriers are having babies, having children, and now we see the first cases of sickle cell disease. There is no way that sickle cell disease should be so common, but if you have one copy of that gene, you have this almost superhuman resistance to malaria. So we don't want to be starting to tinker with the gene pool, because we don't, we're getting into science fiction territory, but it could be a very nasty end product. Was there one more question down here? Yes.
Just a really quick one, going back to your comments about genetically modified organisms, ag biotech. And the gentleman here had posed a really good question around glyphosate and GMO and Bayer, and you talked about Monsanto and Syngenta. What is the role of chemistry or small molecules in terms of its harmful effect, including in ag relative to CRISPR technology or the gene editing that Dow and DuPont and the rest of them are doing today? How would you contrast the three?
I think, with a small molecule, if you're trying to engineer or change the properties of a plant, small molecules are going to spread. Maybe they're going to impact plants that you're not trying to target because they're going to get blown in the wind. You can't box in a small molecule. Whereas in a CRISPR gene editing experiment, you're just fixing the DNA. So I think that there's nothing to spread, really, unless somehow plants are just naturally crossing with other plants. But that's something I think you can control. The spread of a small molecule or a Roundup or whatever the chemical might be, that's not possible. There's growing work about marrying the two. So CRISPR will allow us to do some things, but maybe we want to use chemicals to somehow turn CRISPR off if it's getting too active or something. So I think there'll be some marriage there. And we didn't get round to a question, but we can talk about it offline. You've got the experiment going on in the Keys now with taming mosquito populations. There's a lot of interest, mostly academic at the moment, because but using a genetic phenomenon called a gene drive that could, in principle, provide a sort of a poison pill to mosquito populations that would knock them out, maybe eventually here. Certainly looking at this in Africa, where the mosquito that transmits the malaria parasite doesn't appear to have any value in the ecological system. And if we were to remove it, no one, at least right now, seems to be able to suggest that there's going to be a downside. I'm sure that's not going to be easy, but or proven, but there's always a downside, right? But that's the theory. The problem, I think, goes back, we've touched on it in other questions, is you try telling an African country or government or activists that, we're some big American company and we're just going to come in and we'll use this, sci-fi CRISPR tool to knock out your mosquito population and cure malaria. Their first reaction is going to be, this is just more Western control over my land. We've been exploited enough. Go back to where you came from. But we want to cure malaria. You're losing 500,000 children a year from this disease. We can help you. So right now, I don't think that conversation is really being had, and I don't want to get people too excited. Gene drives still have a lot of work to figure out how that would work. But it is, one of the fun things about this CRISPR story is, and we've seen several examples this evening, is science fact and science fiction, that it's starting to blur. And so maybe 10 or 20 years from now, perhaps, I don't know how bothered people are about the mosquitoes. We're getting bitten by the pool today, so mosquitoes are a problem. But it's a more serious problem at the other tip of the state, where they're transmitting diseases. And so maybe CRISPR will provide a solution to that. We'll see.
Well, if you thought Miami was popular, now wait till we eradicate mosquitoes, and it's going to be really popular. And so we didn't have much time to talk also about velvet eugenics, Julio, which is this idea that the super rich will be able to actually create superhumans. Higher IQ children, and I think that's
Can we knock that one quickly on the head? Because I think whenever we talk about CRISPR, there's a couple of chapters in the book about this whole slippery slope argument. It's great that we can tackle these genetic diseases, but I'm not interested in that. I want hair. I want I want blue eyes. I want
higher IQ.
I want a higher IQ. This quickly becomes it gets very silly. No respectable, and probably not even many cowboys, would start messing with genes that control the brain development or intelligence. Where would you start with intelligence? Can you name me one scientist who could tell me how, just on the back of an envelope, you would tweak a gene or fix a gene to make somebody smarter? I'd love to see that here at the Faena Rose Forum. And they should be pelted off stage if they think that. You'd have to modulate thousands of genes. We have 20,000 genes in our body. Half of them are expressed in the brain, at least. nobody would know where to start. So I think that is essentially a non-starter.
Okay.
But unfortunately, in other forms of health care and reproductive health and fertility clinics, there is the beginning of that slippery slope. We're seeing it. So, just because one can rationally say this will never work doesn't mean that somebody with more money than sense is going to have a stab at trying it. But I'm not too concerned about intelligence. I'm concerned that somebody might want to try it.
Well, I think some people might be rather pessimistic about our political environment being lending itself to the international cooperation that might be required for nations to come together and agree on some of these rules.
Well, it's interesting that, as I mentioned at the beginning, the new science advisor, and this is the first time that we have a science appointee in the cabinet of the president. Eric Lander is probably the leading genomics expert on the planet.
Okay.
I mean, he led the Human Genome Project. He's built the Broad Institute in Boston, taking big money from Eli Broad, the LA-based philanthropist who died recently. He's a polymath. I'm half joking with friends that, on day one, his first press conference, maybe he could stand in the White House press briefing room and just finally lay to rest the lab leak hypothesis. Or maybe he buys into it. But either way, whatever side he comes down on, he would come down on it with conviction, and he would explain it eloquently. I mean, so we've got a real scientist right there in the cabinet now. He's got 3.5 years to make a difference. So let's hope he's successful.
Is CRISPR an example of the way that the scientific community more broadly is opening up to the idea that you can share this kind of knowledge more ubiquitously? Yeah,
that's been a success story. I mean, partly because the reagents and the cost of running an experiment are very trivial. But there's a non-profit in Boston that, for free or for very little money, shares all these reagents. So the moment that David Liu, not this guy, David Liu or whoever, publishes a groundbreaking paper, they send all of their materials to this non-profit, and they become the Amazon clearinghouse to ship these materials around the world. And my journal, the CRISPR Journal, we're publishing papers from Europe, from China, Taiwan, Korea, Australia, submissions from India. I haven't published too many papers from India yet, but it's a truly international science. It's exciting.
Well, coming out of a pandemic, that's a lot of reason for optimism, potentially, that environment is
Well, CRISPR's been, the last chapter touches on,
COVID.
COVID, exactly. CRISPR scientists, including Jennifer Doudna, have been looking at ways to, take their CRISPR expertise and apply it to COVID. So there are new COVID diagnostic tests that have been developed. Because this was so brand new, they're not quite ready for prime time. They're not going to replace the tests that we've all had, the nasal swab and the PCR tests and so on. But I think for the next pandemic, good news, they probably will be ready. And the dream of some scientists is that CRISPR, because it's a scissors that originally evolved to attack viruses, and we've been under assault from a virus for the last 18 months. Could CRISPR be the magic bullet that provides the therapeutic to end all therapeutics? Yeah. The jury's still out on that one for reasons we can talk offline, but people are desperately really working on that. So, there's going to be another. It may not be a coronavirus pandemic, but there'll be another pandemic at some point. I hate to say it, but there will. Because we know that the warning signs were all over the place before the last president took office, and they were ignored. So we'll see where CRISPR ends up in our fight against our viral nemesis.
Well, that's both a very pessimistic and optimistic tone to end on, I suppose. I'm actually going to meet a doctor later who's going to edit my genes to make me just half as smart as you are on the subject of genetics. What a pleasure. Kevin Davies, thank you so much for joining us. Thank you all for coming. Good night.